Hi, my name is Dr. Konrad Wagstyl, and I'm going to be presenting transcriptional cartography to integrate multiscale biology of the human cortex. The aim of this work is to bridge the different scales by which we measure cortical organization, from the nanoscale of genes and cells through to mesoscale layers and columns, and then zooming out to the MRI accessible scales of regions and areas, and ultimately the larger scale atlases and brain networks. And one of the challenges of bridging these scales is the disconnect between the way that in vivo structure is measured and post-mortem maps. So, for example, we have an MRI myelin map. We can get measurements for, in theory, every point on the cortical surface in large numbers of individuals. But when we are comparing these two post-mortem maps, these are generally quite sparse. So this is a fantastic resource from the Allen Human Brain Atlas, where there's thousands of samples within the cortex, but these are done at discrete sample points. And the way this has tended to be analyzed in the past is to parcellate these data into 50 or 100 different regions, which has a limitation that you're imposing anatomy on the samples and doesn't match the dense, comprehensive nature in which the structural function might be measured in vivo. So the aim was to generate spatially comprehensive cortical transcriptomic maps and using the Allen Human Brain Atlas. So we took the 6 donor MRIs, mapped where the samples were to the cortical surfaces, interpolated the values, smoothed them, and then averaged them to generate an average map for each of the 20,000 genes. This is an example of the p-valve map. One immediate advantage that you can see is that for each of these maps, you can also generate the gradient, the rate and direction at which expression is changing for each of the 20,000 maps. We next sought to validate these maps. And so to do this, we did this first at the microscale, looking at cortical area marker genes, so known genes that exhibit clear boundaries of expression. And the best studied border here is V1, V2. So we took the Glasser version of V1, V2 and mapped this to our atlas and compared it with 4 known human marker genes, SYT6, TLE4, PCP4, and PENK. If we look at the expression maps for each of these genes, they exhibit a clear peak in expression within this V1 region on the cortical surface. And if we look at the gradient maps, which is this next column, we can see large gradient changes of expression along this V1, V2 boundary. We can flip this analysis round and do it in a data-driven fashion and say, across all the 20,000 genes, if we rank them by the magnitude of their gradient along this border, where do the marker genes fall? And for the human markers and for macaque markers as well, we see high expression gradients expression relative to all the other genes. So really within the top percentile or 2 for these known marker genes. What this validation enables us to do is say for any given border, can we suggest some marker genes that haven't previously been discovered? So looking at a different cortical boundary along this sulcus between the parahippocampal gyrus and the inferior temporal cortex, taking these 2 areas from the Glasser atlas, this boundary falls along the sulcus, so we can identify this same sulcus within the ISH data. And looking at the available ISH genes, we ranked them based on their expression, their gradient expression along this border, and taking 2 that exhibited marker type features, so aerial marker features in one direction and 2 in the other direction. So we can see that HTR2A goes from low expression in the entorhinal cortex to higher expression in the temporal cortex. And then the bottom 2 are going from high expression to low expression, all with gradients along this boundary. When we look at the independent ISH data, we can see that each of them expresses the pattern predicted. So low expression to high expression. This is the same sulcus that we were looking at on the cortical surface, and high expression to low expression. And then when we do a columnar-based analysis, averaging the intensity within the columns, we see a statistical boundary within this sulcus in the direction predicted by the dense expression maps. So we can validate that these putative aerial markers show strong evidence of being area genes in the ISH data. We then zoomed out to the macroscale patterns and did 4 independent validations of the gene sets. So one was layer 4 marker genes. These are genes that are specifically high expression within layer 4 of the cortex, and compare this to the layer 4 thickness map of big brain. And what we see is that there's a strong spatial correlation between layer 4 thickness and where layer 4 marker genes are highly expressed. The null test is a spin test accounting for spatial autocorrelation. The second comparison we did was looking at bet cell marker genes. So these are genes that characterize these giant motor cortex layer 5 neurons. And we compared these against fMRI-defined motor areas. And we can see that the expression of these bet cell markers is much higher in the fMRI-defined motor areas than elsewhere. So linking cell markers to fMRI through this transcriptomic atlas. The next comparison was comparing myelin basic protein expression with an MRI-defined measure of myelin, so the T1, T2 ratio. And we see that there's a strong overlap between a gene-level description of myelin and the structural MRI-defined metric. And then the final example was linking disease gene sets to disease-related change in cortical thickness. So taking ApoE, and the ApoE4 variant of ApoE is associated with a high risk of developing Alzheimer's disease. And so using the OASIS dataset, we measured cortical thinning in Alzheimer's disease in individuals with this ApoE4 variant, and found that where you have more ApoE gene expression, you see more cortical thinning. So having done these validations, we then sought to look at the whole 20,000 gene landscape and characterize brain areas by their bulk expression pattern. So for each gene, we had a Z-score for each cortical vertex. We took the absolute, so to capture where the extremes were, and averaged this map for each of the genes to give a single bi-vertex measure of transcriptomic distinctiveness, how extreme on average the gene expression is. And when we map this to the surface, you start to see some familiar regions coming out as having particularly distinctive expression profiles. So we see V1, we see the parahippocampal gyrus, we see the temporal sulcus, the central sulcus, the pre and post central motor and sensory areas, as well as the anterior cingulate. We can also look at the gradient of this bulk transcriptomic atlas and find areas where the expression is changing particularly rapidly, but also the orientation of gene expression change. And one thing that we noted is that when we look at the orientation of cortical folds, this is closely aligned to the orientation of transcriptional change. So what we mean is that when you move from a gyrus to the adjacent gyrus, that's the direction which transcriptomic signature is changing most rapidly on average. Having characterized the bulk, we finally sought to break the whole 20,000 genes into groups with shared expression patterns. So we used weighted gene co-expression network analysis, WGCNA, to group genes into modules. And what's nice about this is each module is associated with a set of genes and also a spatial expression profile and gradient. So we can do these macroscale annotations, but also these gene set annotations on the same module. So we ran a whole set of enrichment analysis looking at different types of cortical organization, from macroanatomy like cortical thickness through to functional fMRI-defined networks, and then going down through the scales, layer marker genes, cell marker genes, parts of cell compartments such as the synapse or the nucleus. And then we also looked at patterns of development. So are these genes that are co-expressed spatially in the adult highest expression in early or late development, and also in what types of fetal cells and compartments. There's a lot of different enrichments here to be looking at, but we're just going to zoom in on two modules, M12 and M15. So if we look at M12 first, it's correlated with cortical thickness, has high expression in the limbic regions, particularly high expression in the genes of M12 are enriched for layer 2 marker genes, particularly neuronal marker genes, and this is primarily driven by a synaptic type signature. There's a perinatal peak in developmental expression and is also high enriched for fetal developing neurons. So there's a very synaptic neuronal picture within M12. M15, on the other hand, has an occipital peak in expression closely with the visual cortex. And while it has a neuronal signal, this is primarily driven by a nuclear signal. So it's nucleus rather than synapse. And rather than a perinatal peak like M12, it has a much earlier fetal peak of development. And the less strong signature in the developing fetal neurons and more in cycling progenitors. Having looked at these different scales of healthy organization, we sought to assess whether these modules were patterned for disease risk genes. And what we found is that M12 and M15 in particular had a really strong enrichment for disease genes. And ASD, so the diseases we looked at were ASD, autism spectrum disorder, schizophrenia, developmental delay, and epilepsy, so genes that confer a high risk of developing these disorders. And interestingly, epilepsy was significantly enriched in this M12 synaptic gene, but the autism genes were split between M12 and M15 significantly. So we have a module that's very synaptic and another module that's very nuclear, M12 and M15. And when we look at the Go enrichments for these two different modules, we see that the M12 has a very high neuronal communication enrichment score, whereas the M15, this nuclear gene, is a gene expression regulation module. And this recapitulates previous analyses of ASD genes, where they found that there are these two functional groups of genes within the risk genes for autism. What's nice is that we've captured this distinction just based on splitting the genes according to their spatial patterning. And then finally, if you look at the brain span derived trajectories of these genes, we see this M12 has a perinatal peak, whereas M15 has this much earlier in development peak of expression. Because we have these spatial maps for each of the modules, that we can then keep zooming out and look at other types of pathology within ASD. So this is a study that looked at the number of differentially expressed genes in a number of different cortical areas and identified that the visual cortex has a very high number of differentially expressed genes, whereas other areas, particularly in the frontal cortex, have much lower numbers of differentially expressed genes in ASD. And when we compare this spatial pattern to our two M12 and M15 ASD modules, we find that it's M15, the gene expression regulation module, that most closely recapitulates this pattern that you see of number of differentially expressed genes. Finally, moving all the way to in vivo MRI, we can take the ABIDE dataset, quality control it, and harmonize it for different site effects. And we find that the relative changes in cortical thickness found in ASD, particularly with a peak in the visual cortex and lower in the frontal cortex, most closely recapitulates this M15 module expression pattern. So this gene expression regulation module that relates to the number of differentially expressed genes in different cortical regions might also be related to the pattern of cortical thinning or thickening in ASD. So to summarize, we have these vertex-wise expression maps and gradient maps for 20,000 genes that are available to use and download. They contain biologically rich gene sets, these modules that we've identified and annotated for macroscale, microscale, developmental, and disease effects. And they serve as a resource to link across neurobiological scales, from the nanometer, from the protein-protein interactions, all the way through to centimeter scale folding patterns and functional networks. And with that, I'd like to thank you for listening to this talk. Thank all my collaborators on this project for their different contributions, statistical, analytical, and scientific as well. And finally, I'd like to thank the funding from the Wellcome Trust that has supported me.